yesterday I was in Philadelphia, and often I give talks, of course, to audiences that don't know my work at all, and so there I can give a talk at like 10 minutes notice because I can just give a standard talk. But of course it would be a little bit insulting to most of you, most of whom I actually recognise, to just tell you stuff you already know. So I thought I would try not to do that, but the result is that this will probably be rather shambolic, so I guess I'm rather pleased that we've already had some entertainment that was slightly more structured. But anyway, here I am. So what shall I talk about? Well, I decided that perhaps it would be useful just since the things that I can be most confident that you don't know are the most recent things, just to talk about stuff that's been happening recently and stuff that may happen shortly in the Methuselah Foundation and in related work. Well, I thought I would start with just by telling you how things went towards the end of last year in the Methuselah Foundation, because it was really rather good news. A number of the things that I'm going to tell you now have not actually been publicised in press releases and stuff like that yet, but that's sort of all happening about now, so I'm not really giving away any secrets. But it's all very new news anyway. The first thing that happened was that, somewhat by accident, we got into a situation that very rapidly accelerated the influx of relatively small donations into the foundation. Essentially what happened was that as many of you will probably know, we have an ongoing challenge pledge from Peter Thiel, the co-founder of PayPal, who gives us 50 cents for every dollar that we get from anywhere else. You know, that's been going okay, but it could always go better. And one of our existing medium-sized donors, a guy called Mike Cooper from Texas, said he'd like to give us another $25,000 last month, in December, and that he would like to do it in a manner that leveraged it more, that made more of a difference than just $25,000 might do. So he said basically, I'd like to use it not as a straightforward donation, but as a sort of booster of the challenge pledge that already exists, turning it from a 1-for-2 pledge into something better. And in fact what we decided to do was turn it into a 2-for-1 pledge, basically, so that after this pledge was in place, then anyone who comes along and gives us $1, we actually get a total of $3 rather than only a total of $1.50. And, well, it worked miracles. We exhausted his 25k in a week, or thereabouts, and someone else came along who'd given us a few $1,000 some time ago and said, I'd like to do the same thing, this was such a good idea. So he gave us another 25k and we did the same thing. And then we also were able to use a donation that we'd been pledged last summer from Ryan Scott, whom you may know. Ryan Scott is .com, well, multimillionaire anyway, who is very interested in these sorts of, in transhumanism in general and life extension in particular, and who pledged us $100,000, and we use that in the same way, and that's almost gone now as well. So we're going to try and keep this sort of thing going. It seems to be a very good way to encourage more donations. But the biggest things that happened towards the end of last year were a couple of much larger things. First of all, a guy called Bill Liao, who is the well, he's kind of one of the founders anyway, of a European competitor to LinkedIn, a company called Zing, that some of you may have heard of. He decided to give us more than $500,000, spread over 6 years, but that's okay, I'll take it. And furthermore, it's not in dollars, he's giving us it in Swiss francs, so it might actually be worth something. So that's pretty good. But the best piece of news of all was that Peter Thiel, who has been, as I say, giving us these this matching money when we get money in, decided to do a bit more last Christmas. He gave us a rather nice Christmas present. The current deal is that during each calendar year, he'll give us a maximum of $1 million, and so we are out to get $2 million in that year in order to get the whole million. And we'd only actually got about 2/3 of $1 million, so we only got about 1/3 of what we need to get that whole million in. But he said, fuck it, have the whole million anyway. he reckoned we'd done well enough.
What did he say exactly?
Well, something along those lines. So yeah, he was sufficiently pleased with how we'd been getting on that that was what he decided to do. So we, well, the long and short of it is we had a rather good December. And the reason I thought I would start by telling you all that is because it has resulted in most of what I've been doing since. Of course, now that we have this money in, we can spend it on research, on getting more of the sense plan actually going, getting experiments done that are not being done. And of course, that's my job, you know, as Chief Science Officer of the Foundation, to actually make sure that the right professors are in a position to do the right work as soon as possible, and generally get them, get the money there, get people installed and so on. And of course, to a certain extent, I've been able to do this, to do the first stages of that before the money had actually materialised. I spent most of last year, in one way or another, creating small heads of steam around the world, you know, getting various professors to know that there might be some money to do particular things that they might be interested in doing, but not being able to tell them when it would be. And so there's only so far you can go with that, of course, without really pissing people off. So of course, the money having actually materialised, now there's an enormous amount of work to do to sort out exactly what's going to be done, whether the actual lab work is going to be done by students that are already in the relevant labs or people that we might parachute in, sorting out things like the intellectual property assignments and God knows what like that thankfully are not principally my job. So it's been a lot of work, but the upshot is that it looks very much as though we will be able to initiate at least 3 and perhaps even more projects this year, over and above the 2 big projects that we've been doing over the past couple of years, namely the identification of bacterial genes that can break down things that we can't, and the relocation of the mitochondrial DNA into the nucleus. Both of those projects are obviously continuing and indeed will be growing in terms of manpower during this year. But those of you who've been following this work for some time probably know a fair amount about that, because I tend to talk about them quite a bit in talks. So I thought I would talk a little bit, somewhat shambolically as I've said, about the things that we're going to be, well, that we're currently planning to do some more of. How many people in the room have actually read my book already? A few. That's good, that's good. Okay, so that's good actually, because I'm going to give a bit of background to each of the things I'm going to talk about, and it's background that those of you who actually read the book thoroughly will probably be able to go to sleep for. Another thing I'm doing, which we're likely to do, which is also part of the complex anti-cancer strategy, is to do with stem cells in the blood. Now, if we were to develop a drug that could go in and delete the genes for a gene therapy construct of some sort, that could go in and delete the genes for telomerase so that telomerase couldn't be turned on, and it only went to the cancer cells, cells that were already cancerous, that would be great, in that cells that need to have a bit of telomerase turned on, which includes the stem cells for all of our continuously renewing tissues, like the blood and the gut and the skin and the lung, they would be okay. Unfortunately, a very, very common tactic that cancers use to resist and escape certain drugs of all sorts, not just small molecules, also genetic therapies, is they figure out ways just to destroy the drug or to exclude the drug, stop it being taken up in the first place. So you'll have a minority of the cancer that just won't see the drug, and that's bad enough. Which means that I've always felt that this whole approach of deleting telomerase, and of course deleting the genes for alt as we find them, will require us to be preemptive and actually go and hit all of our cells, not just the cancer cells. So that means we're going to have a problem. If we did that and didn't do anything else, we wouldn't die of cancer, but that's because we wouldn't have time to die of cancer, because we'd have died of not having any blood sometime previously. So the only reason that this whole approach ever seemed reasonable is because of the existence of, or the possibility of, stem cell therapy, of the ability to put cells back into these various tissues, stem cells back into these various tissues, so that the tissue can remain immortal and can continue to produce differentiated cells of the various sorts, various blood cells, skin cells and so on, even though each individual stem cell in the tissue is not immortal. Okay, so there are, as I say, only a few tissues that we have to worry about. Blood, skin, gut, lung. Blood, ostensibly, is the easiest one here, because, of course, replacement of stem cells in the blood is something that we've been doing medically for rather a long time. That's what a bone marrow transplant is. There are things that make bone marrow transplants not as effective as we would like, and there have been, again, some recent advances that have really helped us to become more confident, more optimistic that we can do this. One thing was reported out of Stanford just a couple of months ago. A group found a way to remove the stem cells from bone marrow in a way that didn't involve radiation. This is really important because radiation, of course, it's bad for stem cells, but it's bad for other cells as well. So it's not the sort of thing you want to do if you can possibly avoid it. And this paper, this splendid paper, reported a way of depleting the stem cells out of tissues just using an antibody to a protein that was being expressed on those cells. This is a much gentler approach, much less likely to have major side effects than radiation. So it's a major breakthrough for a whole bunch of reasons, but this therapy is one of the reasons. The process of replacing these stem cells in the bone marrow has to start somewhere. If we are taking even a mouse, or let alone a human, and we want to make all of the stem cells in the bone marrow telomerase knockout, then we can get some stem cells out and delete the telomerase genes and check that we've made the right modification and everything. But in order to get the stem cells back in, we have to do more than just inject them. We have to ensure that those stem cells go into the bone marrow and actually they engraft. And bone marrow transplants are okay for people who are already sick. In other words, whose bone marrow stem cells are already dying for whatever reason. But if the bone marrow cells think they're fine, it's just that they might be at risk of becoming cancerous some decades in the future, then it turns out it doesn't work so well. Stem cells that are happy tend to like to stay where they are in what's called the stem cell niche, and they just don't get out of the way and allow other stem cells that we may have injected to replace them. So this trick of essentially killing the stem cells and depleting them using this antibody is going to make a big difference to the ability to replace stem cells. So partly because of this new advance, and partly because of a couple of other advances in the telomerase field, one thing that I've decided that we need to fund is seriously careful studies on the ability of stem cells that do not express telomerase, that do not have a telomerase gene anymore, to go in and behave just as if they did have a telomerase gene. And you might think, well, why shouldn't they behave the same as normal? I mean, telomerase is only being expressed at this very low level in stem cells anyway. Suffice to say, there's a lot of literature out there that has been purporting to cast doubt on that assumption, that has suggested that there may be other roles that telomerase has over and above lengthening of telomeres. Now, I personally think that a lot of this work has been somewhat overinterpreted, but it remains to be seen. So this is partly a case of just seeing whether the whole, this particular component of the anti-cancer strategy that I've called WILT is going to work at all, but also presuming that it is going to work, it's a way of actually taking it forward in mice. We'll be determining a lot of things like whether the stem cells need to be introduced into the mice when they're young, and in other words, when the bone marrow has not become aged in some other way. To the extent that that might not work, we'll need to determine what aspects of the stem cell environment are somehow inadequate. And the good news is that there's one particular laboratory in southern Germany that's been working in this general space for a few years, and I have good relations with the professor, and he's very interested in taking a slightly different tack in the direction I've just been describing to you, so as to find these things out. So that's the second thing we're going to do. Let me see what I'll talk about next. Yes, another area of sense that's completely separate from cancer is the elimination of cells that won't die. So of course cancer is a problem of having too many cells, but cancer is a problem of having too many cells because the cells are dividing like crazy. They're also dying like crazy, but they're dividing even more crazily. That's what cancer is. There are other problems that are caused by cells that are actually not dividing, but they're not dying either, and they're accumulating slowly as a result, and because they're being brought into existence by other means. And they get in the way. They cause various problems just by being there. Probably the best example of this, the most serious example, though it was only recognised to be so in the past decade at most, is the immune system. In the immune system we have a wide range of different types of white blood cells that are involved in having different functions in protecting us from infections and different cancer, as I mentioned earlier. And they have obviously a large number of different things to do. There's one particular type of white blood cell called a cytotoxic T lymphocyte. A CD8 cell is often called because that CD8 is the name of a protein that these cells express on their surface. That is the main problem in respect of this accumulation of unwanted cells I mentioned. Here's what happens. Some viruses that we get are what are called persistent, which means that we get this infection and the immune system brings it nicely under control and there are no symptoms, or any symptoms that might be are suppressed, completely suppressed, but the immune system does not succeed in completely eliminating the virus from the body. The virus hangs out latently in one or two places. There's a particular family of viruses called the herpes viruses, which are particularly bad at this. And within the family of herpes viruses, there's one virus called cytomegalovirus, which used to be considered to be completely harmless, completely uninteresting from a medical point of view, but which actually is really important for this. Cytomegalovirus clinically doesn't present with any obvious symptoms except in people who've got advanced AIDS or other really severe problems with their immune system. But it seems to be the #1 reason why you have these cells called CD8 cells accumulating in old age in most people. Because you see, most people have CMV. Most people are infected with CMV from an early age. The way it seems to work is that these CD8 cells, which are specific to CMV and are involved in controlling it, divide, and they do control it. They basically get rid of a lot of the virus, but not quite all of it. And they divide some more, and they get rid of some more virus. And every time that the virus tries to have another go at expanding and generally causing us trouble, it gets beaten back, but it gets beaten back by another round of division of the same family of cells that are specific to this virus. And so what seems to happen is a sort of somewhat variant form of what's called replicative senescence in vitro, the concept that I'm sure many of you have heard about, discovered by Len Hayflick 50 years ago, whereby cells end up, as we now know, due to telomere shortening, getting into a state where they can't divide anymore. Now, in the immune system, there's a lot of cell division that goes on, and for that reason, telomerase is actually turned on when it's needed. But probably as a sort of secondary anti-cancer strategy, cells in the immune system, especially this particular type of cell, CD8 cells, don't like to do that indefinitely. They get into a state where the sort of stimulus that would normally make them proliferate and turn on telomerase only makes them proliferate and not to turn on telomerase very much. So they get a bit of telomere shortening, and sometimes you get, and eventually you get into a state where they won't divide at all. They will neither divide nor turn on telomerase. There are various ways we might deal with this problem. One is to turn on telomerase for them, stimulate their telomerase. And actually that's a method that's being explored by a very eminent and prominent professor in this area, Rita Efros at UCLA. I'm a little bit worried about that because it seems to me that this is somehow subverting what I think is likely to be an anti-cancer response, and this may have side effects in terms of lymphoma really rather rapidly. So I'd be rather in favour of simply getting rid of these cells. So then there's one thing I haven't told you yet, which you'll know if you've read the book, but which is pretty simple, which is what's wrong with these cells anyway? Okay, they're not dividing, but other cells can divide instead and carry on, keeping our immune system happy. Problem is that as you get more and more of these cells that aren't dividing, there is an inhibition of the proliferation of other white blood cells. Essentially, there's copy control, there's copy number control. There's there's a system in the body that keeps the total number of white blood cells of various sorts reasonably constant. So that if there's a whole bunch of accumulating useless ones, that leaves less room for useful ones. And there's now a pretty good consensus that this plays an enormous role in the progressive decline of function of the immune system during old age. So we'd really like to fix it. The very best solution would be to actually eliminate the virus, to find some way to do what the immune system is not succeeding in doing and get rid of this virus. But the immune system is very, very, very clever, and we are not that clever yet. And the cytomegalovirus in particular is also very clever. It's a very big virus with a couple of hundred genes, and we really don't know half of all the tricks it uses to hang out and not be eliminated. So we may find ways to eliminate it, but it's not looking like that's an approach whose time has come. So second best is to get rid of these cells that are getting in the way. And, well, there are a few... There's been some good news in that area in the past year as well. Essentially, these cells have been progressively better characterized. At first, it was only possible to identify them by virtue of the fact that they had a bunch of things on their surface that a lot of other useful cells also have, but there was one gene that they didn't have on their surface that the useful cells do have. So in other words, it was a negative definition of characterization of these cells. And it turns out that that's a really tricky thing to use as a target, as a way of actually identifying them clinically, as a way of going after them and making them die or killing them in some way. What you really want is things that they're expressing that useful cells like them are not expressing. And that's now been found. There's a combination of two particular genes, one or other of which are sometimes expressed by useful cells, but both of which are not. So that's going to be rather handy in developing therapies to fix this problem. There are also improved techniques to do something called suicide gene therapy, which is essentially a system whereby you introduce a virus into the body that will go into a lot of different types of cell, and this virus encodes a gene that's really bad for cells and just kills cells. You would think that would be a bad thing to do to people, but at this point it is. But it's a technique that's actually been used in biology, in model organisms, quite a lot. Because what you can do is you can arrange that the protein that's toxic is only expressed if the virus that you've injected goes in cells that are expressing some other particular protein. Essentially, you put what's called the promoter region of some other gene upstream of your toxin, of the gene for your toxin, and that means that the toxin only gets expressed when the other gene is also being expressed. And there are, of course, elaborations of this sort of system that can be used so that it only turns on when there's two genes being expressed, or something like that. So the idea is that you can actually be pretty indiscriminate about where these viruses that are suicide packages will go, because they will do no harm in places where you don't want them to do harm. They will only blow up and kill cells that you actually want to get rid of. So this is, conceptually a nice, simple idea, and as tends to be the way with biology, in practice it's quite messy. But again, progress is being made in this area. So the upshot is that there's a good chance that we're going to be able to fund a project starting this year that will get a good deal further towards the goal in mice of eliminating these clonal expansions of what are called anergic, essentially broken CD8 cells, and the hope certainly is that this will play a large role in rejuvenating the immune system. There's one other thing that we want to do that is also, we're pretty clear, required for truly rejuvenating the immune system, and that is to restore the size of a very important organ in the immune system called the thymus, which is, for whatever reason, something that shrinks throughout life and actually gets, even by the sort of age I am, down to a very small, like maybe 10 or 15% of the size that it was in early life. And it's believed that this has also a rather large role to play in the increasing dysfunction of the immune system. So we want to regrow the thymus as well. There are a few labs that are looking at that. There's one particular lab that's looking at that and is also looking at the problem of getting rid of the anergic cells. And so that's the lab that I'm currently most interested in funding to take this whole thing forward. Yeah, so that's three things that we're doing. Let's see, how long have I got? I'll tell you one more. I'll tell you one more, which is amyloid. Now, you probably all think you know what amyloid is. It's the stuff in the brain that happens in accumulates in Alzheimer's disease, and it probably plays some role in exacerbating the progression of Alzheimer's disease. There's still actually, to be perfectly honest, a good deal of controversy as to whether that's true, whether it really is part of the damage, or whether it's actually harmless, or even in some people think it might be protective in Alzheimer's disease. But for a bunch of reasons, we can say that we ought to get rid of it. If it's protective, then it's protective against something else that we're also fixing independently in the sense plan. So it's definitely an important sense target. Now, amyloid in the brain in Alzheimer's disease is the topic of some of the best news of the year in biomedical technology. News that hasn't really got out yet because it's a bit commercially sensitive, but I'll tell you anyway. About 9 years ago, a company in San Francisco called Elan Pharmaceuticals discovered and published in Nature, I think it was, that if you make a mouse so that it gets Alzheimer's, which mice don't normally do, you can then get rid of the amyloid in the mouse brain, that accumulates in the mouse brain, by vaccination, basically, by stimulating the immune system so that it gobbles up the amyloid. This was a bit of a surprise because, of course, amyloid is a self-protein, a protein that the body actually makes naturally. So you'd think that, like any protein that we make naturally, the body would be tolerant to it. And, of course, that's the problem. The body is too tolerant to it. But there are ways to manipulate the immune system to be slightly less tolerant to something than it otherwise might be. And that's essentially what was done. It was a vaccination to make the immune system think, well, this is a bit foreign, and to gobble the stuff up. And there was preliminary data showing that this actually did cause an improvement of the cognitive function of the mice. So it was obviously a pretty high-profile paper. But more than that, the people who were working on it obviously knew that this was important to move to clinical trials as soon as possible. And, well, it is said that the FDA didn't cut any corners. But all I know is it got the mouse results it went from mice to phase 1 clinical trials and phase 2 clinical trials faster than I've ever heard anything go. And a good thing too, because obviously Alzheimer's is quite bad for quite a lot of people. So anyway, the trial started in probably 2001. And unfortunately, it was a failure. It was a failure in what some people might think was actually the worst possible way, though in a lot of ways I don't think it is the worst possible way. Namely, it had bad side effects. I think the worst possible failure is something that sort of, you spend 10 years doing it and it just doesn't have any effects either way, and you've just wasted 10 years. This, at least, the side effects were found within like 1 year. Side effects only affected about 5 or 6% of the patients, but that was quite enough because the side effects were quite severe. It's basically neuronal inflammation that they got. So one could argue, well, they moved to clinical trials too fast. But actually, it could have been worse because they indeed had moved to clinical trials too fast in the sense that they hadn't thought through the likely consequences of the particular manipulation of the immune system they were doing, the particular type of vaccination, which is called active vaccination, essentially putting the stuff that you want to be gobbled up into the brain and letting the patient develop their own antibodies and develop their own mechanism for getting rid of the stuff. There's an alternative type of vaccination called passive vaccination, which involves doing the actual creation of antibodies not in the patient, but in the way that we normally create antibodies for scientific work, in rabbits or whatever, usually rabbits. So, and then injecting into the brain not the amyloid substance that's supposed to stimulate the immune system, but rather putting just the antibodies in. And that can sometimes be just as effective as a stimulus to the immune system, but it stimulates slightly different bits of the immune system. And this is where I'm saying that, in some sense, the rush to clinical trials may have been a bit too much of a rush, because everything that I've just told you in the past couple of minutes was already known, that the bits of the immune system that are activated by the original type of approach, the active vaccination, are bits that were known to have a risk of causing this inflammation in this inflammatory reaction, whereas the bits that are stimulated by the other sort of, by the passive vaccination were pretty much known not to have this problem. However, that's all water under the bridge, that all happened, and because they figured out very quickly that they really ought to have used passive vaccination, and because more or less everyone agreed with them, they managed to start another trial pretty quickly, using the new protocol of passive vaccination. At the same time, incidentally, a whole bunch of other groups have been looking at other variants, other ways of doing active vaccination, but with tricks that stop the inflammation and things like that. But none of those have got far enough to be really reporting yet. But here's what's happened with the second trial from Elan, who are incidentally a very small biotech company, well, fairly small, but they partnered with one of the biggest pharma companies in the world, Wyeth, for all of this work, and so the trials have been large. So they went to phase 2 clinical trials. Phase 1, as I'm sure you know, is just a very small thing for safety, and phase 2 is sort of doing the safety question properly and also having a bit of a look for efficacy. Now, clinical trials are, like any experiment, you have to do them in such a way as not to be biased by the results. Not to have some sort of feedback where you might influence the result of the experiment by thinking, by what you expect, so to speak. And that's why most clinical trials are done in a manner that's called double-blinded, where the investigators don't know who's getting the drug and who's getting the placebo that's for the control group. And of course the patients don't know that either. But in clinical trials, sometimes you get a drug that's so good, so unequivocally good, that ethically you'd really better know about it pretty early on. And so there are rare examples where enough information is coming back, just anecdotally, that trials get what are called prematurely unblinded. And the data are analysed at an earlier stage in the process than was originally intended. And if it is indeed confirmed that everything is just completely wonderful, then the trial just gets abandoned and the drug gets approved, and there you go. Brilliant. I can't tell you that that's happened with this drug. That didn't happen. But it's only one step below that. What's happened is this. In the phase 2 trial, which is supposed to complete in about April, I think, last year in about August, no, earlier than that, about May, they had an interim peek, as they called it. They had a bit of a look at what was going on. And as I say, this is phase 2, which means predominantly it's about safety, but they are gathering data on efficacy as well, just to see what they see. And, well, they didn't just terminate the trial and just go from there, because it's only phase 2, not phase 3. But what they did do is, they started phase 3 once. They started it back there. Well, they started the process of starting it, and actually it is now started. That is almost as rare as abandoning a trial and just approving the drug. It's fantastically good news. Now, because it was an interim peak and the phase 2 trial is still going on, they have to finish it properly. They haven't unblinded it or anything. That means they can't go public about this. So one only knows about this if one reads the press releases very carefully and/or one goes to the right conferences and talks to the right people. But, well, the long and short of it is, a lot of people are still, of course, not wanting to count their chickens before they're hatched, and rightly so, because clinical trials go bad late in the process rather often, but it's looking damn good. So why am I telling you this? Well, I told you I was going to be telling you about the sorts of things the Methuselah Foundation are going to try to fund this year, or at least may try to fund. The thing is, amyloid is a more general concept than Alzheimer's. There are other tissues in which amyloid accumulates, and in particular there are other tissues in which amyloid accumulates slowly and progressively during aging. There's one important tissue that, in which this happens, which is the pancreas. turns out there's a different type of amyloid, predominantly composed of a different type of protein than the main component of Alzheimer's amyloid, which accumulates in the pancreas and which has been shown quite definitively to contribute to the progression of type 2 diabetes because it kills islet cells. So you probably know type 2 diabetes is a process in which initially the problem is not in the pancreas at all, but elsewhere in the body, with insulin being unable to do its job as well as it should. This is the concept of insulin resistance. The pancreas initially compensates by making more insulin, and that's why if you've got high insulin, it's actually, you might have good glucose tolerance, but you actually have reason to be worried, whereas what you really want to be is someone like me who has perfect glucose tolerance despite having virtually no insulin at all. The second phase of, well, the later phase of type 2 diabetes is where it starts to be like type 1, because the pancreas maxes out, basically, and starts getting to a point where the islet cells just, they die of stress, so to speak. So you actually have a depletion later on in type 2 diabetes of the ability of the cancer to make enough insulin, and that's why type 2 diabetes accelerates. So this amyloid that I've just mentioned seems to play a part in that last process. And people just haven't looked at the corresponding approach. They haven't looked at an immunological approach to get rid of this stuff, this what's called islet amyloid. And they probably haven't done so because nobody was quite sure yet whether it was going to work in the brain, in Alzheimer's amyloid, in humans. And you know, there were other approaches that were perhaps going more effectively, more promisingly in against type 2 diabetes, for example. So my feeling is at this point that probably this work is going to happen fairly soon, but that now is the time when enthusiastic professors can be found who will be interested in pushing this work forward faster than they might otherwise do at this point. There's another type of amyloid which is even more neglected and poorly understood, which derives from a protein called transthyretin. Transthyretin is a protein involved in, well, thyroid hormone transport, not surprising. But it accumulates as amyloid in the heart, and actually in a number of other tissues as well, but the heart seems to be a place where it matters particularly. And it seems to be really bad for really old people. So apparently there's a lot of what is called senile systemic amyloidosis in centenarians, and the heart, as I say, is the tissue that's most affected in terms of loss of function. In supercentenarians, which is people who have reached the age of 110, it seems to be fantastically common. There's a group based in Los Angeles called the Supercentenarian Research Foundation, who are working to study what goes wrong in supercentenarians and how they manage to live as long as they do and so on. And they have got to the point of being able to organise and perform autopsies on a few of the very few people who actually live that long. And more than half of those people appear to have died of senile systemic amyloidosis. So this is definitely not something that we can ignore if we want to seriously do damage to aging. And therefore, again, it's critical that we start to develop an immunological approach, an immunotherapeutic approach to transthyretin amyloidosis with the same ideas in mind. There are some things being looked at by the 1 or 2 labs that work on this stuff, but they are a good deal less likely to, they're a good deal more likely to have side effects, put it like that. So I'm very keen to promote work that very much follows the protocols that seem to be getting so far in Alzheimer's amyloid, and that's another thing that I hope we'll be able to fund this year. And I've got a few other ideas of things we might fund that are slightly further down my list if I get another $1 million tomorrow. But for the moment, I think I'll stop there. Thanks very much. Go ahead, Ralph.
So, one obvious question, I mean, now that you're beginning to get some funding, are those research scientists who previously might have expressed skepticism beginning to shift in attitude, or is that not a visible effect at this point?
Yes and no. The scientists that I'm mainly talking to with regard to actually getting involved are not the scientists who are essentially not at all the scientists who have disparaged science in the past. By and large, the scientists I talk to who are best placed to do these sorts of work are not gerontologists at all. They're people who specialize in other areas that have application in other biomedical fields. Whereas the people who've disparaged science have been all people who would call themselves gerontologists and who therefore think they have an axe to grind when it comes to how to actually postpone aging. However, when I say yes and no, I have been rather amused by the fact that as word leaks out that we've got a bit of money, I do get unsolicited emails from friends in the gerontology community asking how to apply for it. And some of those are people who haven't always been quite so complimentary about it.
Albert, can you tell the group where the researchers are located around the world?
Yeah, sure. Okay.
And then also a little bit about SENS West.
Oh, yes. Okay. Well, why don't I start with SENS West? SENS West is our next conference. So, as many of you know, I have been running conferences in Cambridge in the UK every alternate year for some time now. The first one was in 2003. The most recent one was just last September. And they've been very successful, I'm pleased to say. They've brought together a pretty unique spectrum of scientists who have really enjoyed educating each other, and also plenty of gifted amateurs, shall we say, people like you, who come along obviously to learn the science and also to meet each other. It's a really good crowd. And during the 2005 meeting, a lot of impatience was expressed with the prospect of having to wait another 2 years for all of this. So pressure was put up on us to try to organise something in 2006. And in particular, to do it not in Cambridge, UK, but somewhere that was more easily accessible to people who live on this side of the world and who don't have money to burn. And so that's basically what we tried to do. And we had a couple of false starts, but in the end, what materialised, not in 2006 quite, but early in 2007, was the Edmonton Aging Symposium. One of our stalwart volunteers, Kevin Perrott, who is a PhD student at the University of Alberta in Edmonton, put together, pretty much on his own, a conference that was very much modelled on the SENS conferences. It was only 2 days long in about 3.5, but it was very much the same sort of thing, and it was a complete triumph. So that really validated the idea that we ought to do these things more often and more geographically widespread. And rather splendidly, the opportunity arose for us to do another one this summer. So on June the 27th through 29th, there will be a conference at UCLA, very much along the same lines again, 2.5-day conference, covering all the sorts of things that are covered in SENS and in the SENS conferences, and you're very welcome to come along. The registration fee will not be completely exorbitant, and it's going to have a slightly interesting structure. We've arranged that the 2 main days of the conference will be solid science, very much the way that the SENS conferences have always been, but that will be preceded by an opening session on the Friday evening, the 27th, which will be focused on public policy. And I will have a lot of biologists and technologists, but also people who work on Capitol Hill, people who work in sociology, and we're going to work very hard to make that an extremely high-profile event that gets a lot of media exposure and a lot of attention from policymakers and politicians and so on. We are going to make that opening session free, absolutely open to the public at no charge, which is a good way to get a lot of people. We have booked the largest hall in UCLA, which is Royce Hall, the concert hall that seats 1,800 people, and we are determined to fill it. So yeah, be there or miss something big. But in terms of the locations of the research, yes, so at the moment we are in the somewhat unfortunate position that we are a registered charity only in the US. We are working hard to do something about that. There is a major initiative going on in the UK and another one in Canada to get charitable status in those places. But the nature of charity law is such that each charity is a charity only for an individual country. However, that does not require us to fund research only in the US. We can spend the money where we like. Most of the money we've spent so far has been in the US, and it probably will continue to be for a while, but by no means all of it. The money that's being spent in the US at the moment is mainly on the lysosomal enhancement project, the project of finding microbes in the soil and in other environments that possess the ability to break down things that accumulate in our bodies and cause things like atherosclerosis and macular degeneration. That has been focused mainly in Phoenix, Arizona, a group there that now consists of 2 full-time PhD students and 3 or 4 volunteers. We also have a very talented student working on that same project in Houston at Rice University, and we've just recently started a couple of small collaborations with people working part-time on this project, one at Columbia in New York and another in St. Paul at the University of Minnesota. Outside the US, the main project that's happened so far is what we call mitosense, which is the idea of relocating the mitochondrial DNA to the nucleus in order to make it more protected and basically therefore make mutations in the original mitochondrial DNA harmless. This is something that was not my idea, unlike lysosense. It was first thought of over 20 years ago, but progress was distinctly sluggish in actually making it work, and for no particularly good reason. And I think I'm very happy that I've been able to make a bit of a difference to that, to get work to start happening at an accelerated rate in that field, because it's the first field that I started getting interested in way before I had come up with the whole grand sense plan. All right, so so far so good, and I certainly felt at that point, back in the early 2000s, I guess, and until fairly recently, that this was going to be the way in which we would not completely, but sufficiently completely solve the problem of stiffening of connective tissue during aging. However, it was dealt a bit of a blow by the clinical trials that the company that had the patent for this compound tried to perform. They performed a number of phase 2 trials on various clinical endpoints involved in high blood pressure. And, well, at first I was basically of the view that the problem was they were trying too hard to get results too quickly. They were being driven too hard by commercial pressures, because high blood pressure is something that's extraordinarily difficult to get a drug to treat effectively, or to be seen to treat effectively, because of the placebo effect. In other words, blood pressure is so affected by the nervous system that if you think you're getting a drug, the placebo effect of the things that happen to your system, to your blood pressure, the placebo effect is much bigger than it is in for a lot of endpoints. So if they'd looked at the lens of the eye, for example, if they'd looked at that, then I think they might very well have got statistically significant results in clinical trials. However, that, I'm afraid, is not the whole story anymore. Looking really closely at the results that they got, which, I mean, they did, to their credit, persist and do trial after trial, it's become pretty hard to conclude that that's the only problem. I think now that there's a good chance that the problem is that the type of cross-link that ALT711 breaks is simply not as common in old people as it is in old mice or old dogs. And there are some theoretical arguments that would indeed predict this. Essentially, we know that the type of cross-link that ALT711 is supposed to break, namely alpha-dicarbonyl linkages, is a very fragile linkage that will spontaneously break quite a lot. So one might expect that the abundance of this particular linkage would actually level off over time in a sufficiently long-lived species, so that it would go up during, let's say, early adulthood, but eventually the amount would reach an equilibrium state where spontaneous disruption of these linkages was happening at a rate equal to the formation of new ones. If that's true, then what we would expect is that in late adulthood and in old age, the amount of that stuff would be overtaken by the amount of other cross-links that were formed at a slower rate and therefore had a lesser abundance in early life, but which were not tapering off because they were more stable and they weren't falling apart at all. And we now know that there is at least one, probably two, very weird cross-link species that are indeed of just this sort. They are not formed as fast as alpha-dicarbonyl linkages, or we don't think they are anyway, but they are a good deal more stable, and therefore they may generally be the main problem. So at this point, my focus is not so much on LC711 or on things that might also break alpha-dicarbonyl linkages, and a couple of other companies have been looking at similar things. My focus is on looking for drugs, or indeed other approaches perhaps, that may be able to cleave glucosamine, the main one that I'm talking about here. And this is a pretty tall order. There are some ideas out there for how we might do this, some pretty radical ideas actually, but they are very much on the drawing board at this point. So I would say that out of the 7 sense strands, it could be argued that this one, uniquely out of all the 7, has actually gone backwards in terms of how far, where we've got with it over the last few years.
In regards to the cells that don't die, if you can eradicate them, would you see an emergence or an aggressive reoccurrence of the viruses?
I think I skipped over an important point. Yes,
Yeah.
Thank you very much for this. So so of course there are cells that are specific to one virus, cells specific to another virus. That's how the immune system works, by being very specific to a particular antigen. The cells that we see accumulating are specific to this thing, CMV, but there are also, at the same time, a population of cells that are specific to CMV and yet are not of this character, that are still perfectly capable of dividing. So the belief is that those are the cells that are keeping the virus under control. Therefore, if we only remove the cells that are anergic, that are in this non-dividing state, then we will not be removing the CMV-specific cells that matter, and so everything should be fine. The immune system's reaction to CMV is very well honed, because so many of us have it and we've had it a long time. And so actually the effect on the decline of the immune system that results from the accumulation of the useless cells seems not to actually be manifest in a diminished ability to control CMV. The cells that are still working okay and are CMV specific seem to be really aggressive, and they sort of crowd out. They do more of the crowding out of the cells that are specific to other things and that haven't been so trained.
Right.
At the back. Go.
Where are those cells living? I mean, are they actually in your bloodstream, or are these in your bone marrow?
Not bone marrow, no. Bloodstream and spleen and lymph nodes, basically. The same place as your average white blood cell.
Okay. I mean, the other possibility would just be bloodletting. I mean, you could just force the blood to replenish itself on a more rapid basis.
Yeah. So a variant on that is actually indeed being looked at. What we think we might do is not so much actually make the whole of the blood do so, but rather do some sort of scrubbing. In other words, put the blood back, having removed cells that were in this bad state, without removing all the cells that we'd like that were not in bad state. It might be pretty dodgy to do too much acceleration of blood turnover, because what you'd do if you were just doing it indiscriminately is you would deplete the populations of memory cells against all the other infections that we've had. And for that matter, you'd deplete the population of other cells that, well, memory cells are probably the worst, that you'd have in terms of a side effect as a reduction of the efficacy of the immune system. So it'd be best if you could put back the memory cells and indeed all the other cells that are perfectly okay, and only scrub out the cells that are broken.
I'm only interested because I've heard of studies which are, of course, well publicized by interested parties that say that you actually can, you're healthier later if you donate blood than if you don't. Of course, it's kind of self-selecting in the sense that people who are healthy tend to donate more blood. So.
It's self-selecting, and also, I mean, there are other effects. For example, it's pretty clear that giving blood is a good thing for men to do, and possibly for postmenopausal women, just to keep the iron down. But, only up to a point.
Aubrey, which will be the last organ that will be able to fix, like, will be the brain?
I think it probably will be the brain, to be perfectly honest. The brain is not intrinsically different from other organs in the sense of things going wrong there that are qualitatively different from things that go wrong anywhere else. Or at least we don't think it is. We don't know whether just knowing a lot is bad for you.
Is it a joke?
But I mean, one can argue that at the gross level, everything else can be replaced wholesale. You know, if we have problems with getting an immune approach to getting rid of transthyretin actually working, so that the heart does accumulate this type of amyloid and it does suffer, then, you know, we can always grow new hearts in pigs or whatever and stick them in wholesale, and that, you know, of course we've got some way to go to be able to do that, but we may very well find that that wholesale organ replacement approach will end up being easier in some organs. For the brain, that would rather defeat the object.
Although there are some people.
There's always one.
Volunteer round?
All right.
I guess this kind of links to the last question, maybe a little bit more longer term, but what is your take on the role and feasibility of neural prosthetics as a means to life extension?
It's not really a field that I'm expert on. I mean, the general concept of non-biological augmentation of the brain is, or indeed non-biological replacement of the brain, uploading and variants thereof, is an area that a lot of people have thought about a lot more than me. All I would say is that I'm pretty conservative when it comes to futurist thought in general. I even get slightly uncomfortable when people call me a transhumanist sometimes. And I'm certainly, you know, my instinct is that I rather enjoy being made out of meat. But, but despite that, when I think about it, it's occurred to me over the years that I actually don't have much particular emotional attachment to what my brain's made out of. So if we did have some sort of system for, especially if it were a system for gradually replacing biological neurons with non-biological ones, in such a manner that you know, neural function of all sorts was maintained throughout, then, and of course you'd end up with a brain that was rather more robust because it wouldn't rely on having a continuous supply of oxygen, and it might be backable up and things like that, then I could go for that. And I have a feeling that when we fix aging properly, people are not going to be terribly keen on being hit by trucks, even at age 800 or whatever. So there may be enthusiasm for this sort of approach. All right. Oh, you want to?
Yeah, your research investment strategy seems to be to do several different research projects at the same time. Have you considered the possibility that focusing on one particular one could get you maybe not commercial success, but a stronger solution quicker, which could then end up refunding you and driving things quicker? Right.
Yeah. We have what I would call a mixed strategy in that respect. There is value in and of itself in working on the broad swathe of sense strands, simply to give ourselves a distinctive, you know, place in the biomedical world. Really, you know, people look at us and they don't say, oh, that's another Alzheimer's charity, or that's another atherosclerosis charity, or whatever. So that has value. However, what you say is absolutely true, that any particular strand that might be able to be pushed through either directly to commercializability, or for that matter, simply to big credibility, you know, the discovery that somewhat radical approach that other people might have disparaged has actually borne fruit. You know, that sort of thing would certainly make a big difference in terms of credibility, and therefore very possibly in terms of the rate of donations. So the projects that we are pushing most strongly is definitely defined dynamically, very much in response to what happens, in terms of how soon we may make progress. However, counter to that, we take the view that the ones that are furthest away from progress there's also a good reason to fund them at high priority, because otherwise they will continue to lag and we'll end up, you know, in a position where we fixed most things, but we're not actually seeing much actual benefit. So, there are lots of conflicting pressures on us in this way, and that's why we take a rather hybrid strategy.
This is sort of a question out of left field, whatever. It's probably not too closely associated with aging, but the virus, the threat of death, the flu virus.
All right, SARS, yeah, avian flu, what?
Well, yeah, what's the largest threat of virus to come out
any type of, not the avian flu, but smallpox.
Right.
Smallpox, yeah, but that has killed
A lot of people.
a lot of people in the past, and it's sort of a potential to do so in the future. Is there something?
Well, my view is here that it's something that will be solved as an economic and social consequence of solving aging. At the moment, 98%, I would say, of the reason why serious infectious disease are still a serious problem is because vaccines are not profitable. Vaccine research is a major black hole. It's just not much, not a good way to make money quickly compared to what the alternatives available to big pharma. Now, big pharma can get away with that, or shall I put it another way, nobody else steps up to the plate and does something about that, simply because at the moment in the developed world, not many people die of infectious diseases, except people who are old enough that they're really dying of aging. Like, you know, obviously lots of elderly people die of flu and so on. So there's just not enough public pressure there to create the economic pressure. This, I think, is one of the many things that's going to change when suddenly we aren't all dying of other things.
Will that remain sort of a risk in terms of?
I think it will always be a risk, yeah. I mean, there will always be an arms race between the microbial world and the human world. No question about that. I mean, I think even if after we upload, we've still got to worry about computer viruses. Yeah, but sure. I mean, yeah, that's never going to go away. But it's a question of whether we can, whether our ingenuity and approach to these things is going to be able to stay one step ahead of the problem to an extent that it really does remain a negligible cause of death. And, well, it does become a negligible cause of death, shall I say, and I think that's likely to happen when we actually put our mind to it.
In your theory about combating aging, how do you factor in like chronic inflammation over the years and free radicals and stress responses?
Well, the short answer is I'm afraid you're going to have to read my book because there's no because in order to give you an answer that's really convincing and concrete, I would have to give a very long answer. But if I give a short answer that you may find a little bit too superficial, free radicals are precursors of accumulating damage. Free radicals themselves are molecules that are very reactive. So they don't accumulate in the body, they get created and then very quickly they react with something and they may cause damage to DNA or damage to proteins or whatever. The focus of the whole approach that I take to combating aging is to focus on what's accumulating rather than on what's causing it to accumulate. And there are reasons for that come down essentially to the practicalities of the complexity of the human body, the fact that it's easier to avoid unforeseen side effects that way. Inflammation, the situation is slightly different. Inflammation is a response by the body trying to make things better, and essentially the main reason why inflammation is a major role in aging is because lots of aspects of aging have some characteristics in common with infection. So essentially what the immune system does, or not just the immune system broadly defined, what it does is it responds to certain aspects of aging, like the accumulation of cholesterol derivatives in the artery, for example, as if it were an infection. Now, the reason we have the inflammatory response is because an infection, when it's got rid of, the important thing is to get rid of it quickly. And once you've done that, any damage, any collateral damage you may have done to yourself by really going hard after the infection, you can repair it at your leisure. So inflammation is bad for us, but in its appropriate context, it's only bad for us temporarily. The problem comes when something that is not going to be eliminated by the inflammation is nevertheless triggering inflammation. Because then the inflammation fails, and it tries harder, and it tries harder, and it carries on doing more and more damage to the surrounding tissue. That's why aging exhibits a lot of inflammatory characteristics. Now, if we ask why, okay, yes, that may be the reason then, but it's still a problem, right? The thing is, well, no, because what we want to do then is simply to remove the problem that's causing the inflammation. So in the case of cholesterol in the artery wall, for example, that's part of the Lysosense project, to go in and find enzymes that can remove these variants, these oxidative modifications of cholesterol that our existing natural enzymes can't remove. Once you've removed them, everything starts working okay again in the middle of, in the original source of the inflammation. That goes away just as if it were an infection, and then the inflammation recedes and regresses just as it does after a real infection. I need a beer.